Alright, welcome back. This video is going to be looking at the cell membrane, its structure and its function. Goes along with section 5.1 and 5.2. And the cell membrane is extremely important, as we looked at in our Agar Cube lab. It regulates what comes in and what goes out of a cell through a process called diffusion, which we'll be looking at very extensively on Friday and next Tuesday. The makeup and the action of the cell membrane is very important to understand to be able to understand diffusion. It's made up of many different things, as you can see from this computerized picture of the cell membrane. These blue kind of squiggly lines are proteins, and this blue balls down here are phospholipids, and then we have the fatty acid tails right there. Alright, so let's look a little bit more into structure and function of the plasma membrane. So I think a picture's worth 1,000 words. This is a picture of the plasma membrane. And you can see there's a ton of stuff that make it up. We've got the extracellular matrix, the collagen fibers, the actin filaments, glycoproteins, glycolipids over here. So many different things to look at. But in general, we want to do the first thing first. What does the plasma membrane do? Well, it surrounds the cell. It's the boundary, the inside and the outside. And it controls what substances enter and leave. It's like a wall, a fluid wall or a gate. It controls what goes in and what goes out, and also is in charge of a lot of processes in which the cell interacts with things on its exterior. The things that it's made of is it's mostly phospholipids, major component phospholipids. That's what these little balls with two tails like balloons with two strings looks like. They have embedded proteins, so these purple things are proteins. And steroids, so cholesterol, when we're talking about steroids, is a form of lipid, cholesterol, and carbohydrates, these green things. The carbohydrates are very important for cell signaling and for the cell to interact with things on its outside, sense another cell is close by, or if a pathogen is coming in to harm the cell. And these proteins kind of float around, as you see, in a sea of phospholipids. So think of these phospholipids as like the water in a pool, and all these proteins as like balls, beach balls, floating around in the pool. And that gives you an accurate kind of representation of how they kind of float through the plasma membrane. And because, we've got these dotted solid proteins in this sea of kind of fluid phospholipids, a lot of scientists say the phospholipid or the cell membrane is kind of like a fluid mosaic. It's fluid, meaning that these phospholipids shift around and are kind of like a fluid, movable texture, and then they have proteins floating in there. So the most important part of the plasma membrane is the phospholipid bilayer. Bilayer means two, so two layers of phospholipids, one stacked on top of each other. And first of all, from the last time we talked about it, a phospholipid is just kind of a substance that's made up of a phosphate group. So up here, phosphate functional group, a glycerol, so this middle part, just like a fatty acid or a triglyceride has a glycerol, and then two fatty acid tails down here that can be saturated or unsaturated. Saturated, no double bonds; unsaturated, one double bond or more. So unlike a triglyceride, there are only two fatty acid tails in a phospholipid. And the important thing to know about phospholipids is the head is hydrophilic. It's polar, likes water, dissolves in water. So see this phosphate group, very polar. It will like or dissolve in water. The tails are hydrophobic, hates water, non-polar. So we have a hydrophilic, likes water part, and a hydrophobic, doesn't like water part. Polar, non-polar. What happens then when you put two phospholipids together? Well, if this is the outside of the cell and this is the inside, the water-hating parts, or the non-polar parts, the hydrophobic parts, will try to scrunch together, get as far away from cluster, bunch themselves up against the water, just like hydrophobic R groups. And the tails, the fatty acid tails, will move in towards each other, making a hydrophobic region. The tails, the heads that love water, polar, hydrophilic, will flip outside towards the extracellular fluid, the water on the outside of the cell, or the water on the inside of the cell, the hydrophilic area. So these heads will flip towards the extracellular, intercellular fluid and form this bilayer, one phospholipid oriented right side up, one upside down in a row, kind of like this. And it's these non-polar interactions that keep the membrane together, hydrophobic clumping together, hydrophilic towards the water. So one really important thing about the plasma membrane is its fluidity, that fluid mosaic model, its ability to be kind of like a sea that proteins float around in like a beach ball. But the plasma membrane can vary in its fluidity. So for example, membranes with a high saturated phospholipids, what does that mean? No carbon-carbon double bonds, are less fluid, especially at low temperatures. For example, that is because there's no kinks. There's no carbon-carbon double bonds, so the fatty acid tails can be straight and they can pack together really tight. And it makes it more viscous or more thick, especially at low temperatures. On the other hand, if there's a phospholipid bilayer that has many unsaturated fatty acids in its phospholipid, you'll get these kinks and they'll kind of push each other apart. They can't pack as close together and it becomes more thick or less thick, more fluid. So, for example, in the free response question for your test, cold water animals, cold water, are going to have more of these type. They still need to get oxygen in, carbon dioxide out. And if it's too tightly packed, diffusion is greatly hindered. Can't get oxygen in and out or water in and out. Warmer water creatures will have a higher concentration of saturated fatty acid. In addition, cholesterol helps maintain the fluidity of a phospholipid bilayer, especially in humans. For example, this cholesterol kind of wedged in there spaces out these phospholipids, and it makes the plasma membrane more fluid at lower temperatures, especially in humans. So for example, those same cold water fish might have an increased frequency of cholesterol in their phospholipid membranes because they live in low temperatures. However, at high temperatures, the cholesterol makes this phospholipid bilayer less fluid. So it's kind of a checks and balances mechanism. And a lot of organisms have evolved to be able to modulate their saturated and unsaturated composition of their phospholipid bilayer and their cholesterol composition of their phospholipid bilayer over time based on their environment. It's a very great adaptation evolutionarily, and that has to do with the Golgi apparatus and its ability to process and where it ships those saturated fatty acids, unsaturated fatty acids, and cholesterols. There's membrane proteins that are extremely important. They do a variety of functions. There's over 50 kinds of membrane proteins in the plasma membrane. But in general, there's two broad categories. Integral proteins that are embedded inside of the phospholipid bilayer, like these. They are inside, spanning the phospholipid bilayer. And there's periphery proteins, maybe like this one, that only are on one side, the inside or the outside, or this one. They don't cross over through the phospholipid bilayer. As you can see, they have many different functions. So, for example, as we'll talk about in 5.3, some proteins, membrane proteins, are transport proteins. So, they transport substances from the inside or outside of the cell to the other way. Some are just like tunnels. Some require energy to pump. Other proteins catalyze enzymatic reactions that are important to the cell membrane. Some act as signal receptors, so a molecule might bind with a protein, an integral protein in the plasma membrane, and that protein might send a signal into the cell to tell it to do something different. They're also important in intercellular joining, so two proteins might join with each other to join two cells together in like a desmosome, like we talked about, those junctions in cells. Proteins might be attached to the cytoskeleton and the extracellular matrix, important in changing shape of the cell, or modulating shape. And lastly, we'll talk about in chapter 6, cell-to-cell communication is the job of proteins, cellular proteins. So some proteins on the cell membrane have these carbohydrate chains attached to them and they're called glycoproteins. Glyco meaning sugar, glycoproteins. There's some phospholipids that are called glycolipids. They have the same chains, and what they do are they're used for communication recognition in between cells. So one protein might be a receptor, and when it binds or gets close, fits in the shape of this glycoprotein or glycolipid, it might say like, hey, I'm here, and tell one of the other cells to do something. Right. The most important feature of the plasma membrane is that it's selectively permeable. It decides what goes in and out. So it only lets certain things in. It's selectively permeable. In general, because of this large area in between of nonpolar hydrophobic fatty acid tails, usually only small nonpolar molecules can get through. So water sometimes even has a hard time getting through, because even though it's small, it's polar. But small, like oxygen, like nitrogen, those things can get through just by diffusion. They can diffuse right across the plasma membrane. Larger polar molecules have to go through special gates called transport proteins. Some transport proteins are just tunnels that the certain molecules, if they fit, can go right through. Those are called channel proteins. Channel proteins. They just don't move at all. They're just big holes that let certain molecules through. Others, called carrier proteins, actively bind on to the molecule that needs to be moved from the outside to the inside or the inside to the outside, changes its shape and drops it off on the inside. Even water sometimes needs to go through these transport proteins, specifically these channel proteins, and when they carry water, they're called aquaporins. Alright, lastly, just to review, we've got our plasma membrane made up of phospholipids, bilayer. They have a hydrophobic region in the middle, hates water, made of fatty acids, and a hydrophilic region, the head, made of the not the polar phosphate. They have proteins, integral proteins and periphery proteins that do special functions, some of which are channel proteins, which passively let different substances too big to diffuse through the membrane through. Some are carrier proteins, which actively pump from one to the other. And there's a bunch of glycoproteins and glycolipids, these carbohydrate chains stuck onto the plasma membrane of the proteins that help in cell-to-cell communication and cell recognition. Remember, small, non-polar substances are the only ones that can naturally diffuse through the plasma membrane. Small molecules or non-polar molecules. Sometimes you can have small polar molecules. Sometimes you can have large non-polar molecules, but usually it needs to either be very small or very non-polar. Alright, that's 5.1.5.2. Go through that activity in class, do a case study. We should be ready to go with osmosis diffusion. If you have any questions, just let me know.